Hi everyone, welcome to today's episode on photosynthesis exam questions. I'm going to show you from start to finish how to think, approach and answer the questions to maximise the amount of marks you get. But let's begin. It says, suggest 2 ways in which the ultrastructure of the chloroplast can be altered by high temperatures, and for each suggestion, explain the effect that it will have on photosynthesis. Great. So, this is suggest. So you kind of need to think a bit more abstractly, to say the least, for this. But you don't really need to do that. Because if you reword this question to something that makes a bit more sense, you'll get to the answer very fast. So how I would reword it would be like, okay, in the chloroplast, if high temperatures occur, what can go wrong? That's it. Do you know why? Because high temperatures affects what? Enzymes. Affects membranes. And that causes a lot of problems for photosynthesis. And that will be the answer. So for example, let's cast our minds to the light dependent stage. It is very dependent on membranes, is it not? And so if there is damage to the membranes in the chloroplast, due to these high temperatures, what would that reduce? Light dependent stage. That will be one suggestion and one explanation. Another suggestion I can make is that, there might be damage to the chlorophyll. What does that reduce? It reduces the light dependent stage. That's really it. It's very basic. Just think about where are you in this question. Well, I know I'm in the photosynthesis topic, okay, but where is high temperatures most likely to affect more? Well, the light dependent stage. Okay, well what actually happens in the light dependent stage? Where is it? What are the key components? Membranes, chlorophyll, and so that way you can make your answers. Okay, next question says, what conclusions can you draw from the data in figure 3.2 about the effects of light and the internal structure of a leaf? Explain your answer. So I'm going to read all the information here because I feel like it will be relevant so that I understand what on earth I'm trying to answer. So it says palisade mesophyll cells are an example of a specialized plant cell. These cells are the main site of photosynthesis in plants. A team of scientists investigated the effect of shining light on the upper and the lower surfaces of leaf. That's very important. Now I know exactly what's happening on the rate of photosynthesis. So this is what they're trying to test out. Okay, now we're going to look at the graph. What's on the x, what's on the y-axis, how are the graphs looking like? So it's comparing 2 things, which is the upper surface and the lower surface, and we are measuring the rate of photosynthesis. Okay, so now let's break down the question because it's quite loaded and it's a 4-marker. Okay, so conclusions I can draw from the data, so that's the first thing. And number 2 would be, what are the conclusions about a leaf? What's the structure of a leaf? But you can't just be like, oh yeah, it's like chloroplast, this, and this. No. It means from this data, from this figure, what can you draw about or understand what's within a leaf? That's what it means. So, as you can see, I've got 2 photos, and they are there to serve a purpose. Okay, but I'll get into that. Number 1, the first thing you do in a graph question is just state the obvious. Look at the graph, and if you had to explain it to like a 7-year-old, what would you say? But you still have to use like biological terms, okay? And not completely like a 7-year-old. But, so we know that the upper surface is the dots, and the lower surface are the squares. Now let's look at the graphs one by one. So if I was to take, this is number 1, number 2, okay? For graph number 1, it has a steeper gradient, and also at lower light intensity, right? Lower light intensity, it has a greater or higher rate of photosynthesis compared to graph 2. And that continues and continues until they both plateau at the same point. Do you think that the difference between them is quite significant? So when you actually look at this graph here and this graph here, which one has more of a significant change or difference to graph number 1? It'll be this one, right? And so when you look at that comparatively, there isn't much of a difference between graph 1 or 2 being the upper and lower surface. So that's just an important point to recognize, okay? So on to actually answering the question. Well, we start off by saying a clear description of the graph. You can be like, well, at low light levels or low light intensity, the photosynthetic rate is greater when the light is shone on the upper surface, right? That was the thing we were talking about here. So that's one mark. Another thing you can talk about is how there is no difference in the rate at higher light intensities. So there is no difference comparing graph 1 and 2. As I said, they both plateau at the same point. We've already got 2 marks. And I literally just stated the obvious from the graph, okay? I haven't even gone into the explanation yet. A third mark that you would get is by using figures to support your conclusions. So I can be like, yes, well, by saying there's no difference in the rate of photosynthesis at higher light intensity, I can quote a figure here and a figure there. I have to use the units, right? And that gives me 3 marks. In these sorts of questions, you always are guaranteed 1 mark to use a figure, as in figures as in numbers from the graph, but you have to do it with the units, okay? Guaranteed 1 mark. So already we're on 3 marks, and I haven't even got into the explanation that I want to give you guys yet. So let's see where our fourth mark can come from. So, another thing I could talk about is about the plateau and why the graph is shaped this way. Because at the end of the day, we are talking about plants. We are talking about photosynthesis. There will be limiting factors, right? So here, one can say that at higher light intensities, aka this region, carbon dioxide concentration is a limiting factor. That can explain why the graph looks like that. Okay, it's not going to be light intensity because we have it on the graph, so it's going to be carbon dioxide. Okay, that's 4 marks. But there are other, I would say, not niche points, but some points that most people won't get. And they are these. 1 mark you can say is how to explain why the difference between upper surface and lower surface is so minimal, right? The reason for it is that leaves are thin. That's why we've got this image here. When you pick up a leaf, right, you can kind of see the other side. It's kind of translucent in a way. And it's because they are actually really thin. You know this, if you just try and rip apart a leaf, do you know how easily it can be done? It's because it's thin. So when you use that logic, well, wouldn't it make sense for me to say that light can pass through the leaf from the lower surface to reach the palisade cells, right? So when you look at the anatomy, if you shine it this way, okay, palisade cells are over here. But because the leaf is so thin, okay, that it can just honestly penetrate through. So that's a reason why we can say that the graph kind of looks like that and why there isn't much of a difference between both the graphs. So yeah, a lot of ideas there, but honestly, for 4 marker, it's very easy. You just state the obvious, especially here how 1 mark was for the number and figure, a number and unit, sorry, and others were description, and a bit of explanation. But honestly, with a 4 marker, the world is your oyster. Go crazy. I would say put as many points down because you don't really know which points are going to be on the mark scheme, so you'd rather be safe than sorry. Okay, and that's the end of today's episode. Remember, the two most important exam techniques are read the question first and state all the obvious points. Now, over to you. Try some questions and let me know how it goes.